Okay, Assalamualaikum and hi to everyone. My name is Nurul Fatin Suhabinti Shukri and I'm going to present in a structure and functions in Pecha Kucha style, which is my assignment one. So yeah, let's start. Okay, DNA is the largest macromolecule that represents the genetic material of the cell. In 1953, Watson and Crick proposed the three-dimensional of DNA on data obtained by Franklin and Wilkins. Deoxyribonucleic, or as known as DNA, is the macromolecule that holds the genetic information of living things. In our body, each cell consists about 2 meters of DNA. That's a lot, right? Okay. DNA is made up of two polynucleotide strands. Based on Watson and Crick model, polynucleotide strands twist around each other and forming a shape that looks like a twisted ladder, and it is called double helix. Okay, the polynucleotide strands joined by phosphodiester bond, as we can see in the slide. The polynucleotide strands run anti-parallel to each other with nitrogenous bases. The strands run in opposite direction but parallel to one another. The head of the strand is always laid against the tail of the other strands. This is due to the different polarity that both of them experience. In figure shows, the left-hand strand has 3' to 5' polarity, whereas the right hand has 5' to 3' polarity as compared to the first one. There are two types of nitrogenous bases, which are purines and pyrimidines. The two carbon-nitrogen ring bases, which are adenine and guanine, are purines, while the one carbon-nitrogen ring bases, which are thymine and cytosine, are pyrimidines. Chargaff rules stated that the amount of adenine, thymine, guanine, and cytosine in DNA varies from species to species. In each species, the amount of adenine is equal to thymine, while the amount of guanine is equal to the amount of cytosine. Okay. Adenine always pairs with thymine by 2 hydrogen bonds. Meanwhile, guanine always pairs with cytosine by 3 hydrogen bonds. This creates a difference in strength between the two sets of the Watson and Crick bases. Okay, for your information, guanine and cytosine bonded base pair are stronger due to the 3 hydrogen bond bonded between them, as compared to thymine and adenine base pair, which only consists of 2 hydrogen bonded base pair, as you can see. Okay, next. Apart from that, the DNA helix has shallow groove called minor groove, which equivalent to 1.2 nanometers, and also a deep groove called major groove. And this major groove, which equivalent to 2.2 nanometers across the DNA. Okay, we proceed to the functions of DNA. The main function of DNA is as heredity material. The genetic information stored in nucleotide sequence of DNA helps in synthesis of proteins and polypeptides, and also it transmit the information to daughter cells or offsprings. Thus, the DNA is called as molecular blueprint or thread of life. Next, DNA also important as they create variations in population and ultimately contribute to evolutions. DNA undergoes recombinant at meiosis and occasional mutation which changes in nucleotide sequence. As you can see, there are crossing over between the chromosome that leads to variations. Okay, moreover, I'm sorry, DNA also plays a very important role in genetic engineering, which is recombinant DNA technology. It involves the artificial cleaving and rejoining DNA sequence from two or more organisms to create a recombinant DNA. This technology is employed for the production of genetically modified organisms, GMOs, genetically modified foods, GMFs, hormones, vaccines, enzymes, and also clones. Recombinant DNA also produce insulin, human insulin made by bacterial cells. Firstly, human gene removed from the cell, then insert into the plasmid. Plasmid then insert into bacteria, and bacteria produce insulin as it was one of their own gene produced. This is the process of human insulin in recombinant DNA. Okay, the next is DNA also important in DNA fingerprinting. Each individual has specific short nucleotide repeats which are called as mini satellites. The probability of having two people with the same DNA fingerprint is very small. We can use polymerase chain reaction, which is PCR, to increase the amount of DNA sample. It only requires a small amount of DNA to start with, and even can make copies from the DNA sample that is partially degraded. Okay, last but not least, the DNA also have function in heterocatalytic role. During transcription, only one strand of DNA serves as template for synthesis of RNA. This is called heterocatalytic role of RNA. That's all from me. Thank you.